This is, I want to say, slide 28. You guys are looking for where we're starting. So we went through a lot of definitions on Monday, all the terms that have to do with epidemiology and what study of epidemiology is. And now we're going to talk about a little bit more about disease specifics as they relate to epidemiology. So the patterns of disease, obviously there are some predisposing factors that will determine the course of an infection or the severity of an infection, and these are fairly common sense things. So physical traits. So, for instance, females are more susceptible to certain types of infections because of their reproductive system. They have a shortened urethra, so they have urinary tract infections much more frequently than men do, just because of the physical traits. There are inherited traits, so things that have to do with your genetics. Sickle cell anemia is one of the major examples of that. Climate and weather, you guys are all aware of that. The flu always comes out as the weather gets cold, and that's not a coincidence. That's because of people's immune systems are down, and so there's things about the climate and weather that affect the host, such as the cold weather making your immune system be depressed, but they also affect the bacteria, so bacteria survive better in the colder weather, so forth and so on. Fatigue, and I'll put stress there too, so you guys probably experience that. As you get high stress, you're more likely to get ill. Again, that has stuff to do with your hormones and the chemicals that are released, and as stress is induced or fatigue is induced, and that suppresses your immune system. Age, basically, when you're really young, you get sick. When you're fairly healthy, and then when you're really old, you get sick again. So that's the way things go. Again, based mostly on the immune system. Lifestyle. So if you go out partying every night and you're drinking all the time, you're more likely to get sick. And then chemotherapy. So certain, again, general chemotherapy. Certain drugs that you may be taking may make you more susceptible to other types of diseases. So here's a very generic graph of the stages of a disease. So again, this is different for every type of disease, for every person, for every individual infection. Even within a person, this could shift one way or the other, but this is a very generic region. So there is initial infection when you're first exposed to the microbe, and then there's some incubation time where basically the microbe is replicating and within your bloodstream or within your cells. And during that time, there's usually no signs or symptoms. Okay, so I always go back to the flu because it's the easy one. Usually you get infected with the flu. There's an incubation time where you feel fine, usually 24 or 48 hours, and then the symptoms come on. So although you got infected, the symptoms don't come on for a couple days. Then there's a period where you start to have mild signs or symptoms. And again, this is where the virus or the bacteria would still be replicating. And then there's a longer period where you have the peak of the illness. This is where you'd have your most severe signs and symptoms would come about. Your immune system, or if you're on antibiotics or whatever, starts to dissipate the infection and get rid of the microbe, in which case you get a period of decline where you still have signs and symptoms, but they start to be less severe. And then you have a period of convalescence, which is basically just healing, and at that point you really have no signs or symptoms, but you haven't completely rid yourself of the infection. So at this point, convalescence, again, you have no signs or symptoms, but if something else triggered a reinfection, you still have some remaining microbe left in your body, so you could get a second peak here of signs and symptoms. So here's showing the very wide range of incubation periods for different types of infections. So a salmonella food poisoning type infection, usually you feel that the same day. So if you eat something for lunch that was undercooked, you get salmonella food poisoning, usually that night you're not very happy. Okay, so we're talking 6 to 12 hours of an incubation period before you start to feel signs or symptoms. The flu, 24 to 48 hours after infections when you start to feel the signs and symptoms. And then there's they're basically showing the extremes here. HIV, again, typically 1 to, here it says 8 years, and now with common drugs that can be pushed out even more. The one on the other end of the spectrum from a food poisoning would be leprosy, where you get infected and then you have no signs or symptoms for up to 30 years. And then once it triggers, once you have the signs and symptoms, they're very severe, but you have a long period of no symptoms. Okay, so how do infections typically get spread? So usually from reservoirs of infection. So what that means is these are places where the bacteria or the virus is constantly found. That is a continual source of infection for people that come in contact with it. So the continual sources of infection, if we're just focused on humans first, so like HIV, sexually transmitted diseases, there's always some proportion of the population that has this infection, so there's this continual source of infection just out in the human population. The carriers may have inapparent infections or latent diseases, so again, this is common with STDs, that the person may appear healthy, yet they have sexually transmitted disease that they can pass on to somebody else. Animals can be a source of infection. So rabies and Lyme disease are two of the big ones. And again, we're talking transmission from animals to humans. And so this could be from a wild animal. So like Lyme disease is from ticks. So, out in open field areas, you can get bit by ticks and they get Lyme disease. Rabies can actually happen from domesticated animals. So you can get rabies from dogs. Obviously, they should have their rabies shots. It's not always the case. So you can get these types of infections from both domesticated and wild animals. They are difficult to eradicate because a large source of the infection is from wild animals. And you can't basically treat all the wild animals out there and get rid of the disease, so there's a constant source out there. The third class would be non-living. So botulism, toxin, tetanus, usually you get these not from a person or from an animal, but from an inanimate object. So a lot of times, like for instance, tetanus, if you like step on a rusty nail or something like that, you're always suggesting go get a tetanus shot. You find it in soil, water, food, and these are basically virtually impossible to eliminate. So how do the diseases get transmitted from one source to another, whether it be a human, an animal, a non-living object? Well, there's direct contact. So that basically is close association between an infected and susceptible host. So when we talked about direct contact, we're talking person to person, okay, or animal to person. Indirect is when we talk about an inanimate object that's capable of carrying the infectious organism. So that's the difference between direct and indirect. Okay. And then there's droplet transmission, which is airborne droplets. So direct contact transmission, physically touching somebody that has the infection, and then here's a super gross picture of somebody sneezing, showing what this is the main type of droplet transmission. This is from somebody sneezing in the general vicinity. Yeah.
What's an example of indirect?
So indirect would be like again the tetanus, like from a rusty nail, or anything where you're getting from a inanimate object. So even if someone, let's say someone had an infection and they were sitting at the table, got up, and then you came down and sit at the table after them, and you got the infection that way. So that would be indirect. So direct is close association, so usually, close contact with someone. Indirect is usually the infection or the microbe is left behind and then picked up by somebody else. So here, vehicle transmission. This is getting a little bit more into that inanimate reservoir of infection. So there's airborne transmission. So you can get infected from dead skin cells. So that'd be example, someone's infected, they're sitting at the table, some of their skin cells are left behind and they have bacteria associated with them, and then you pick them up. That's one way. Dust, soil. Usually, this can happen with bacteria, it can happen with virus, it can happen with fungal spores. Waterborne transmission. So pass through food and water. The water can be the reservoir and the vehicle, so meaning that the bacteria could be living in the water. And then if the water is picked up and ingested by somebody, it then becomes the vehicle for infection. And then there's foodborne transmission. Poorly processed, undercooked, poorly refrigerated foods. hands not washed thoroughly prior to food preparation. So vectors are basically, in this case, again, we've talked about vectors a couple times. A plasmid's a vector that carries DNA. In this case, we're talking about a vector as an arthropod that can spread a disease. So basically an insect that can spread a disease. So fleas, ticks, mosquitoes, those are the three big ones. There's two general methods in which these insects can transmit a disease. So there's mechanical transmission, which is basically, let's say there's some dog feces on the ground in your backyard. A fly lands on it and then flies into your house and lands on your food. So it's basically physically picking up the bacteria and moving it inside the house, and then you ingest it. That's one way. Okay. And there's biological transmission, which is like the intermediate host, where the pathogen is actually inside the vector, reproducing inside the vector, and then the vector bites you and transmits it that way. Okay, so now we'll talk a little bit about nosocomial infections. So I've talked a little bit about nosocomial infections. Basically, the definition of nosocomial infection is an infection that you get by being in the hospital. Okay, it's not an infection you get and then you go to the hospital. It's an infection you get while you're at the hospital. Okay. So it's a fairly high number of people actually acquire these nosocomial infections. 5 to 15% of all patients that are in the hospital get one of these infections. Okay. So here's the reason. It's kind of counterintuitive. You think you're going to the hospital to get healthy, and then you end up with a fairly potentially serious infection. Okay, and here's the reasons why that happens. Well, one, you're in an environment that has a lot of sick people in it. So there's a lot of microorganisms floating around. Okay, there's one reason. Another one, there's a compromised host. So a lot of people that are in the hospital are ill for some other reason. So their immune system is already a little bit depressed. So they're more susceptible to infection. So you have people that are highly susceptible to infection and an area that has a lot of infectious material in it. So it's a bad combination. And the third one is the chain of transmission, which basically means, and this is, try, as much as they can, health professionals try to minimize this one. The other two they can't really do much about, but this one they try to minimize. This is basically meaning that a doctor goes into a patient's room and sees a sick patient, and then 5 minutes later goes to another room to see another patient. So there's a physical means by which a microbe can travel from one patient to another. So the combination, the intersection of all those is why there's such a high prevalence of nosocomial infections. Here's a pie chart showing the different types of nosocomial infections. So a large percentage of them are urinary tract infections. Anybody have an idea why that might be?
Catheters?
Right. So catheters basically is introducing the possibility of infection into an area that typically doesn't have microbes in it. Where else? Surgical site infection. So again, basically causing an open wound as a place where bacteria or virus can get in. So here's looking at the percentage of total infections, not by what procedure the patient is undergoing, but what type of bacteria is causing that. So there's these coagulase-negative staph infections. That's about a quarter of all infections. And a high percentage of those are resistant to antibiotics. So that's a very serious infection. Staph aureus. So these are about a sixth of the infections. Again, very highly resistant to antibiotics. Enterococcus and the gram-negative rods are not as serious because they're still pretty susceptible to antibiotics. So MRSA is the big one. That's the staph, the methicillin-resistant Staph aureus. And I've talked about that before. So there's two prevalent strains in the U.S. There's USA 100 strain and USA 300 strain. The USA 100 strain is almost exclusively found as a nosocomial infection. It's almost exclusively the only way you really get this strain is from a hospital stay. Okay, there is a strain that you can get just in the general population, but that's usually a different strain, USA 300. Okay, so here's MRSA patients. and versus what type of procedure they're undergoing, and the total number of patients who receive that procedure. So you see hemodialysis, out of 1,800 people that they surveyed, not surveyed, of the data they had, out of 1,800 people, 800 of them had MRSA infections. So we're talking almost 50% of the people that had this procedure got this infection. Okay, close to 40%, but still pretty significant. Where you see an IV catheter, 1,000 people, but that was out of 16,500, so not as high percentage. But you can see it's pretty significant percentages depending on the different types of procedures. There's the last section we'll talk about for this lecture, the emerging infectious diseases. So these are fairly new diseases. These are the type of diseases I want you guys potentially to focus on for your end of the semester report. Newer diseases that haven't been around as long, they're starting to study more. So these are diseases that are new, increasing in incidence, or showing a potential to increase in the near future. So they could be new diseases that we haven't seen before, or they could be diseases that have been around for a while but haven't been a major problem, and now all of a sudden they're becoming a major problem. So like MRSA would be a increasing in incidence, because Staph aureus has been around and people have been getting staph infections for a long time, but it's becoming more and more serious because they're becoming resistant to antibiotics. Okay, so that would also be considered an emerging infectious disease. So here's the major reasons why there are emerging infectious diseases. So there's genetic recombination that's always happening. So again, we've talked about E. coli 0157, the E. coli strain that picked up the Shigella toxin. So that's always happening. So new combinations of strains are always occurring. Same with evolution of new strains. So a strain can also, just by genetic mutation, can start to take on new traits that make it harder to treat. Talked about this one fairly significantly. Inappropriate use of antibiotics and pesticides, get more antibiotic resistant strains. And then the changes in the weather pattern. So you guys have all heard of global warming and that, and how melting the polar ice caps and all that's going on. But another side effect of the weather patterns changing is that we're having new infections that hadn't been as big of a deal before. They're now becoming more and more prevalent. A couple more reasons. So modern transportation. So a long time ago, before people were traveling to different countries across the globe, a disease would be prevalent in one place and would basically stay there because people weren't traveling and spreading it around. Okay, obviously now we're in a more global society, people are traveling all over the place. And so, for instance, West Nile virus. West Nile virus used to only be contracted on the west bank of the Nile. Okay, and then people started traveling there and bringing it back to other countries, and now you can basically have West Nile virus anywhere in the world. Expanding human settlement is also along those same lines. So moving into areas that weren't inhabited before, and we're finding new different strains of bacteria when people do that. Animal control measures. Lyme disease, again, is a big one. The tick population is increasing, so Lyme disease is also increasing. And then public health failure. So diphtheria, does anybody know what that is referring to? So diphtheria, I can't remember what country it was. Let's say Russia or something like that. So diphtheria is a disease that was basically completely eradicated from the population because we had a very good vaccine, and it was one of the vaccines that children get almost immediately after they're born, within 3 or 6 months after they're born. It's called DTaP, maybe you guys have heard of it that way. That's what the D stands for. And so basically we were, not we, but health professionals were inoculating children with this vaccine, and it was basically gone. And again, I can't remember where this happened, but they basically came to the conclusion that nobody gets this anymore, so we don't have to inoculate for it anymore. And basically they stopped giving the shot. When they stopped giving the shot, they saw a significant peak in the number of people that were getting diphtheria. Okay, so again, it can be a public health failure where we have something under control and then we change something and then it takes over again. And I've showed this before, I'm not going to go over this again, but this basically just shows a diagram of how the swine flu came about by genetic recombination. And this is another example of an emerging disease from the last 10 or 15 years. Any questions? All right, so we're going to move on. I don't want to do a break right now because I think we can probably get through this next lecture in half an hour or so, and then you guys will just have a nice little break at the end.